Okay, I guess we'll get started. Um, Anybody has a question or something you want to talk about? Okay. So last Monday, I guess it must have been, we were, or no, even before that maybe, last week, 2 weeks ago, whatever, we were starting on a topic having to do with some of the unexpected results that came out of the initial production of transgenic organisms. And I mentioned, and I showed some graphs showing that some of the results we were getting really were not what we expected. In other words, you know, when we were studying prokaryotes, and even when we were studying yeast, every transformant behaved like every other transformant, basically. But when we started making transgenic multicellular eukaryotes, so those would be things like plants or mice or fish or things like that, we found that you couldn't rely on a single organism to tell you what the effect of your mutation was really doing. Every transgenic had the potential to be slightly different in the expression of a transgene from every other transgenic. And it took a while to begin to figure out what was going on. Along the way, one of the big points that told us that this was worth studying was the fact that the level of expression of the transgene did not correlate with the number of copies of the transgene. And that was quite unexpected. So it was saying to us, each individual integrated gene behaves slightly differently, or at least had the potential to behave slightly differently. And then in some cases, for example, I showed you some pictures of some petunias, I believe. People found that when they introduced even a sense gene, it sometimes led to the shutting off, or silencing as it came to be called, of both the introduced gene and any endogenous gene with a very high degree of homology to it. So for example, though we put a gene that should have increased the purple pigment in the flowers into the petunias, we found that the flowers were white, and this was a heritable trait as far as could be determined. So there was a lot going on we just could not explain at that time. And after a number of additional experiments, the first thing that we were capable of doing was resolving the differences into two forms of gene silencing. One of them came to be called transcriptional silencing. In transcriptional silencing, the gene is being expressed, at least to the extent that you can see the primary transcripts in the nucleus. But you didn't see the messenger, and you didn't see the proteins. I'm sorry, I'm already mixing these up. That was a description of post-transcriptional silencing. Post-transcriptional silencing is this concept that the effect of silencing happened even after transcription had occurred. So we had transcription in the nucleus, but we couldn't find the messenger and we couldn't find the protein. So that was post-transcriptional silencing. And then we also had transcriptional silencing, where there was simply no detectable level of RNA, either in the nucleus or elsewhere. So what we're going to talk about a little bit today and afterwards is going to be these two processes, some of the typical experiments that have revealed these two processes and the limits of each of the two processes. And at the end, we're going to try to explain them. So in the beginning, I'm not going to be providing an awful lot of explanation. I'm more going to be describing what scientists found. And you can follow along and think for yourself, well, how would I explain that, for example? But at the end, we will try to, I will try to explain some of that stuff and bring the two forms of silencing together, because we do think today that these are actually part of a single pathway, even though they're revealed in different fashions. Okay, so I actually have talked already quite a bit about chromatin structure and how changes in chromatin structure can influence the expression of a gene. We mentioned, for example, that there are ways to modify the histones, that these modifications occur primarily on around the 5' and promoter regions of a gene, and that if the chromatin is modified in a way that increases its packing, it tends to shut off the gene by blocking the ability of RNA polymerase to unwind all those proteins. And then we're also going to have cases where when the gene is meant to be expressed, that chromatin loosens up. And I showed you these polytene chromosomes at one point or another. Anybody want to tell me what a polytene chromosome is? These are these chromosomes found in Drosophila salivary glands. Anybody know that from previous terminology? Sir?
I remember in a previous class, didn't you say that all the chromosomes are linked together or something?
Yes, but not end to end, but rather side to side. So, in other words, if you think of a chromosome in a eukaryote, it's a strand of double-stranded DNA, and then it's got all these proteins around it that wind it up into a much smaller package so it can actually fit into the nucleus. We can't see those with a normal microscope. And, and even preparing it with an electron microscope is a little bit of an art. But long, long ago, Drosophila geneticists saw that the salivary glands of Drosophila had visible chromosomes. They look like these pictures here, where you would see these dark and white bands. These came to be called polytene chromosomes, many-stranded. That's what that means, many-stranded chromosomes. The idea is each chromosome is amplified, is copied, replicated, and chromatinized many, many times, hundreds of times, and then each is aligned side by side with its own kind. So you are amplifying whatever the pattern is on one molecule is now being amplified over 100 molecules exactly in order, you might say. And so all these copies of chromosome 1 look this way, or maybe I should say all these copies of the first homologue of chromosome 1 look this way, and then all the copies of the first homologue of chromosome 2, of the second copy of that chromosome 1 look that way. And then you go to chromosome 2 and chromosome 2A and 2B, chromosome 3, 3A, all of these things. So when they looked at this and saw these dark and light bands, and then they were able to coordinate these patterns with genetic maps that they had created, they could recognize that the dark bands tended to be areas where genes were not being expressed, and the light bands were areas where they were being expressed, according with this type of a staining protocol. And then they came to call this expressed form of chromosomes, of a chromatin, euchromatin, and the unexpressed form, the heterochromatins. Heterochromatin can vary as genes get turned on and turned off, but it's always present around centromeres and it's always present around telomeres, and tends to always be present around places in the genome where there's a transposable element, a repetitive element of some kind or another. The fact that the chromatin differs gave us one possible explanation for why transgenics could differ. A gene could integrate in euchromatin, in which case at least it goes into a place where in theory it's capable of being transcribed. Or a gene could go into heterochromatin, where in theory it's going to be suppressed or silenced in some fashion. And so that was one explanation, but other explanations have come to light over the course of time, and one of them is the fact that just as there are modifications on the proteins that surround the chromosomes, there's also modifications of the DNA itself. There's a process of methylation of cytidine groups that people have identified. And what that means is basically that if you have a cytidine group that looks like this, there will be enzymes that can methylate that cytidine by adding this methyl group here. And by putting that there, it can erase or cloak that sequence from the ability of a transcription factor to bind to it. It's even possible that the transcription factors we are most familiar with, so that would be those that bind to enhancers or bind to silencers, are actually meant to be able to distinguish between methylated and unmethylated sequences. They can bind, they can see the unmethylated sequences, but apparently they cannot bind or see the unmethylated ones. By comparison, DNA polymerase is not affected by this, RNA polymerase is not affected by this, and neither are any of the repair enzymes. It's really limited to the transcription factors that would be binding enhancers and silencers. These methyl groups can be permanent through the life of an organism, or they can vary. On the average, what people have seen is that maybe 25% of all the Cs in a vertebrate or a plant genome get methylated in this fashion. Invertebrates, like Drosophila and C. elegans and so on, don't methylate their DNA. We think this methylation is an additional way of silencing the expression of a gene. A more permanent way, you might say, than, for example, using a silencer binding protein to shut a gene off. Many of the examples of genes that undergo a transition from an unmethylated state to a methylated state are genes that are meant to be expressed at one time and not after that one time. So the classic example that people give is studies of the fetal hemoglobin gene in mammals. In the embryos, this gene is expressed to produce a version of beta globin that has a much higher affinity for oxygen than the adult version of beta globin. It's necessary because it's trying to capture all that little bit of oxygen that's coming into the into the embryo, for example, from the mother. But once the embryo has been born, this protein would actually be deleterious because it would bind oxygen extremely tightly, which is good, but it wouldn't release it, which is bad, and therefore it would deprive the tissues of the oxygen that they really need. So what the genome has to do is silence these fetal hemoglobin genes forever and ever. And so that's done apparently with methylation. We only can say this by correlation, but what we can see is that the timing of the methylation of the promoter regions of these fetal hemoglobin genes correlates with the silencing of the fetal hemoglobin expression. And so we believe that this has to do with, that the silencing that we see is, in this case, the result of this methylation process. Now, one of the things we're going to be trying to distinguish for the moment, or not for the moment, but in the future, is the idea that there are many different mechanisms in the organism to methylate DNA. Not different so much in the sense of the enzymes that are responsible, but different in the purpose of that methylation itself. We've talked about methylation in prokaryotes, where methylation was a way to protect against restriction enzymes. But in eukaryotes, we don't have restriction enzymes. Methylation is only used there as a way of regulating gene expression. And there seems to be, as I mentioned, two different broad kinds of methylation going on. One of them is what we're just going to call maintenance methylation. This is the kind of methylation that ensures that telomeres and centromeres, repetitive elements, and so on are always highly methylated in a way that it shuts off anything that they can do. Later, we're going to talk about a very special form of methylation that also has a big effect on gene silencing. But that is much more gene specific, and it is much more determined by circumstance than it is by a general process. So let me just dig into this whole idea of maintenance methylation first of all. In this kind of a process, what we see is that the enzymes that are responsible for methylating the genome, methylating the telomeres and the centromeres and the repetitive elements, tend to look for specific motifs in the DNA. They tend to look in general for CCGG. In plants, we have CCGG, but we also have CCNGG. In mammals, mostly CCGG. And when this methylase finds one of those sequences, it's going to methylate that second C in each of those sequences. And having it on one strand and then having it on the other strand ensures that that sequence is invisible to the transcription factors that we've been discussing. The process is heritable. It's just like the inheritance of methylation in prokaryotes, at least through mitosis, should we say. Meaning that if you start out with a fully methylated sequence, let's say like this, then when DNA polymerase replicates this portion of the genome, it's going to make two double-stranded sequences, each inheriting an old copy, in other words, one of the methylated copies, and a brand new copy recently synthesized by that DNA polymerase. These are hemimethylated regions, just like they were in prokaryotes. And they have a particular attraction for the DNA methylases, the maintenance DNA methylases of the cell. So when we start with these hemimethylated sequences, DNA methylases will quickly find those and methylate the unmethylated sequence in the opposite strand and restore the fully methylated state to that particular sequence. Okay. Any questions on this? Now, we have developed a number of ways of detecting this methylation. We can't use simple DNA sequencing easily, but people have developed some rather complicated enzymatic processes that can be coupled with standard, for example, Gilbert-Maxson sequencing, and sometimes even with standard PCR processes, so that one can distinguish whether a particular sequence in the genome is methylated or unmethylated. But the older way of determining whether there's methylation in this region of the gene is to use methylase-sensitive and insensitive restriction enzymes. And so that's the process I'm going to talk about, because that's the process I'm going to show you in a subsequent paper we'll be talking about. So what does this all mean? Let's imagine that we have a chromosomal sequence that looks like this. This is a region of a chromosome that we are interested in because of the presence, let's say, of that gene. And these are the locations where these CCGG motifs happen to be. We would like to ask, are any of these methylated or not? Or more importantly, are those that are part of the gene or its 5' sequence methylated or not? So what we're going to do is we're going to use Southern analysis to answer this question. We're going to take genomic DNA and cut it with divide it into two halves, and cut it with two different restriction enzymes. One is we're going to cut with MSP1 that's able to recognize both the unmethylated and methylated versions of CCGG. And then the other is going to be cut with HAPA2. HAPA2 recognizes the same CCGG, but it can only cut the unmethylated version. And by comparing a southern hybridization with DNA cut with each of these two enzymes, we're going to be able to ask which of these sites is methylated and which is not. So let's say we take our genomic DNA and we cut it with MSP and HPA2. And we run it on a gel, we stain it, and what we do is see millions and millions of bands of all the sizes from the highest molecular weight to the lowest molecular weight. We cannot see any differences, even though the enzymes are cutting differently in part. We visually would never be able to see those differences when we're considering the total genome, a digest of the total genome of the organism. But what we're going to do now is we're going to blot that and hybridize it with a probe. In this case, the probe can be the entire gene or just a part of the gene. In the example I'm using here, we're only taking a part of the gene. It's just for convenience sake, but it's possible. And it happens to lie here so that part of this probe sequence overlaps this 2 kb fragment, the distance between this site and this site, and this 1 kb fragment, the distance between this site and that site. Is everybody okay there so far? Everybody understands southern analysis and all that stuff. So now, let's hybridize our genomic DNA that we blotted to a filter with that probe. If the DNA was not being methylated, then when we hybridize with that probe, we're going to reveal two bands, the 2 kb band and the 1 kb band. And so we'll see a pattern like this. And it'll be the same whether we used MSP or HPA2, because there's no difference. I mean, the enzymes may be different, but the sequence of the DNA is unmethylated, so both enzymes can cut it. On the other hand, if this particular sequence was methylated, when we digest the DNA, again, we're not going to see any difference visually, but when we then hybridize with that probe we were using, we would see that MSP, which is able to cut both methylated and unmethylated sequences, will generate those two bands, the 2 kb and the 1 kb, because that probe can independently hybridize to each of those two bands. Right? But when we hybridized when we digested the DNA with HPA2, we see a single band of 3 kb. And from that, we can conclude, by comparing this and this, we can conclude that that middle CCGG pair was uncuttable by HPA2. And so the fragment that was released extended from this site to this site, giving us a 3 kb fragment. Pretty basic. Everybody okay on that, or you want me to go through that again? And we can choose other probes from the same genome, from the same gene, and we can probe additional regions. In general, we are not too concerned with whether or not the sequences of the transcribed region of the gene are methylated or not, because there are no transcription factors binding there, and RNA polymerases doesn't care about the methyl groups. What we are concerned about, if we want to explain whether this gene is being expressed or not being expressed and why this gene is not being expressed, what we're concerned with is 5' sequences. And so you can go as far up as you want to visualizing by southern analysis different portions of this 5' region until you have mapped as many of these methylated sites as you can. Before we go through an example of this, I want to also though point out that this process, this method, has a lot of limitations. Technically, it's exceedingly easy to do, but it only is able to recognize sites where we have restriction enzymes available for cutting them. So we are not doing a complete scan of the 5' region of this gene. We're only really scanning a small region. But in general, if the methyl groups tend to be within about a kb or so of the promoter region, it's likely that even though we are only detecting some of those methylated sequences, there are adjacent ones that are methylated as well and could be explaining the silencing of this particular gene that we're talking about, we're investigating. Okay? Does that make sense to everybody? So we're using some representative sites to conclude something about a region at the 5' end of the gene, and if we see methyl groups at the test sites, you might say, it's likely there are methyl groups of other cytidines in that same vicinity, enough to probably overlap some of the enhancers or silencers for this protein, for this gene. Now, what I want to do is give you an experiment which helped to reveal how this process, which I've described so far in terms of like maintenance methylation, actually can provide us a strange explanation for a gene-specific methylation process, a transgene-specific methylation process. And the experiment that revealed all of this was done using a really weird little organism called a viroid. So I want to first talk to you a little bit about what viroids are, and then we'll go into the actual experiment itself. Anybody know what a viroid is? They don't teach you these in micro, I guess, that's okay. Viroids are a kind of pathogen. They're a weird kind of pathogen. They are primarily found in plants, but there are viroids in mammalian systems as well. They are single-stranded circular RNA molecules, very small single-stranded circular RNA molecules of about 250 to 400 nucleotides. Now that by itself is strange. Circular RNAs are kind of weird, although we now realize that there's more examples of circular RNAs out there than we ever imagined before, but this is a particular class. These viroids can cause growth inhibition of the plants that they infect, but they can only be transmitted by wounding. That means if you crush a leaf in your fingers and then you touch another leaf, you can transmit it. But the viroid doesn't encode any proteins. It never gets packaged in a virus shell. It can never leave the plant on its own the way that a natural virus would be able to. In addition, these viroids have a lot of internal complementarity. So in solutions, they can fold up into structures of stem and loop structures like this, where you've got sort of arms that are base paired and then regions which are not base paired. And as mentioned here, just for the heck of it, they are resistant to radiation, UV radiation, and all sorts of things like that. As I mentioned, it's very important to keep in mind they do not encode proteins. They do not integrate into the genome. They simply replicate in the nucleus when they infect another plant. But the process of replication is one of the things that makes them so weird. What happens is that this circular genome is recognized by RNA polymerase II of the plant, and it begins to copy it in a rolling circle-like fashion. So the same way that lambda late replication occurs the same way that plasmid conjugation occurs, the same way that tDNA transfer occurs, also occurs in the replication of these viroids. But in this particular case, as I said, we're using an RNA template and RNA polymerase to do the work. The polymerase will just keep going around the circle over and over again, generating an end-to-end concatemer of that circular genome. And then that linear molecule that's created undergoes a self-cleavage reaction. What that means is that these RNAs are like, well, if you know what the term ribozyme is, they are ribozymes. If you're not familiar with ribozymes, just think of them as an enzyme made of RNA rather than DNA, that in this particular case is capable of cutting itself into small pieces and then circularizing each of those pieces without the assistance of any protein. We can transcribe these things in a test tube, and those concatemers that are formed will process themselves into circular single copy molecules. Like this. All right, so far. Okay. We're now going to go to a paper where their goal was trying to understand this process of replication, but in the course of doing these experiments that they hoped would reveal what was critical for this self-processing of the viroid RNA, they came across a totally different phenomenon related to silencing. And this is this paper by Vasudeva et al. I don't remember if I sent this to you. Did I send this in this? Maybe not. Okay, well, I'll try to send it as well as a second paper. It's not like I'm going to ask you to tell me things that I haven't said in class, but sometimes it does help to go through the paper and see if you understand the figures and so on, and maybe even understand what their point was when they were making this. So in this particular paper, what they had done is they cloned initially. They did a series of experiments. In their first experiment, to try to better understand how viroids replicate naturally, they first made a cDNA copy, and then cloned that cDNA copy between a strong promoter and a terminator. And when they expressed this in plants, it was transcribed, but it never circularized. And if the promoter was shut off, those RNA molecules never replicated themselves. They said, well, what's wrong? We thought we had everything that we needed here. They tried different versions of the cDNA. None of them worked. So they said, well, if we look at the natural course of events, the viroid first makes a concatemer. So maybe we should be trying to clone a concatemer. So they made a concatemer in vitro by just ligating these independent copies to themselves. And then they cloned that concatemer between a strong promoter and a terminator and introduced that into plants. And when they did, they got the long RNA complementary to the gene they had put in, but then that RNA self-processed into circles that then replicated independently of the transcription process going on with this particular integrated version. Okay? Well, part of the study, anytime you're doing this kind of study, really requires that you make an estimate of the number of copies of the gene you have put into the plant and made a determination whether or not the gene that integrated is really the version of the gene you wanted and not some kind of mutated or scrambled version of that gene. So they had to do some southern DNA analysis on the transgenic plants. Looking at the map of the concatemer, they figured that they would have, let me just see here for a moment. Sorry. Before I mention that Southern analysis, I do want to mention one additional experiment. As part of their process of trying to figure out what part of the sequence was necessary for replication, they made a small deletion, and actually they made a few different deletions in their cDNA copy, and then concatemerized that. The question they were asking was, if we make successive deletions in this particular viroid, do we finally find a specific sequence necessary for replication, assuming the rest of the sequences were not? So they then concatemerized these deleted versions of the viroid genome to make a copy like this, put it behind a strong promoter in front of a strong terminator, introduce that into plants, and it was also transcribed. But it never self-processed into viroids. You never saw circles, and you never saw any kind of replication going on independent of the transcription of the integrated gene. And it didn't matter where those deletions were made. Any deletion, anywhere in the viroids, abolished the ability of this concatemer to process itself. So now, with these two lines of experiments, the fact that if you have or maybe three, you have a single copy, you get no replication and no circularization. You get a concatemer, you get circularization, and you get replication. And then you make a deletion in each copy of that concatemer, and you don't get replication or circularization.
I was wondering, do you know about how long the deletions were? Were they really large chunks or small, or
they were
just,
relatively
one?
small.
Okay.
So I don't particularly recall how big they were, but we're talking on the order of 10 to 20 bases.
Okay.
Okay. So they were trying to scan the length of the sequence step by step in hopes of finding a particular region necessary for replication and self-processing. But it turned out every one of them abolished replication and process. So all they could do is accumulate this long concatemeric RNA. Okay, so they had evidence that a single copy cannot replicate and circularize, that a concatemeric copy can replicate and circularize, and that a concatemeric copy with deletions cannot circularize or replicate. They were ready to think about publishing. So the first thing they did was, of course, as I started to say, they had to verify that the genes they had introduced were the genes they had constructed and had not undergone some kind of scrambling process that would have interfered with the ability of the viroids on their own to replicate. So when they looked at the sequence that they had created, the tDNA sequence they had created, they found that there were HA3 sites which can cut GGCC or GGC 5-methyl-C, but not GGC GGC 5-methyl-C. So they can cut the sequence if the methyl group is on the first cytidine, but not on the second cytidine. And that's fine. They weren't anticipating that they would be looking for methylation, I should say. They only used this enzyme because of its convenience. It would cut each of these units in the concatemer, and it would also make a cut in the 35S promoter that was driving the expression, and it would make a cut somewhere in the tDNA itself. So in theory, when this DNA got in, if it was intact and built and integrated in the same way it had been built in the test tube, when they cut with HA3, they expected a band of 1200, which would be from the last copy of the viroid into the tDNA itself. They expected a 510 base pair copy, which would be the first copy of the viroid going into the promoter. And then they expected a very dark band where each of these internal repeats of 359 bases would migrate. Each of these would go from viroid to viroid, viroid to viroid, and viroid to viroid, okay? So they expected these three bands to be found. And that would cover the entire region that they were interested in, so if it worked, it would tell them that, yeah, the gene had integrated in an intact form. They didn't find that. Instead, what they found when they cut with HEY3 was a single band, and this is when they hybridized with a single copy of the viroid, by the way, okay? So that was their interest, is visualizing these bands, because each band had a piece of the viroid sequence in it, so their probe carried the viroid sequence. Instead, what they found is a single DNA band of about 2,420 bases, which would actually be the link going from this site to somewhere beyond here. Somewhere out here in the chromosome. So how is it possible that when you digest and then hybridize, you find one big fragment instead of three small fragments? The only explanation went back to the fact that HEY3 could not cut sequences that had a second, that had a methyl group on the second cytosine. So it suggested to them that each of these HEY3 sites in this tDNA had become methylated. Okay? But here was the weird thing. This was what they saw when they investigated the genome of, or the integrated copy of the normal viroid, the one that where it was intact, capable of replicating on its own, and so on. When they hybridized to their mutant tDNA, the one that has these little deletions, the one that doesn't lead to circularized RNAs and replication of those, they found the pattern they expected. So methylation was only occurring on the integrated copy that was being, where the viroids were being produced, and not on the integrated copy where the viroids were not being produced. Everybody okay so far? If methylation is silencing transcription, how is it possible that the integrated copy of the concatemer that was producing viroids was able to produce viroids? Why didn't methylation prevent that copy from being expressed, so to speak? Now, to answer the question, what you want to do is you want to picture in your mind the sequence of events that we're talking about. We are looking at a temporal process. The first step of that temporal process is the integration of the tDNA itself. That tDNA would have been unmethylated. It had a good promoter and terminator, so it would have been transcribed, and it would have produced a long concatemeric RNA. That long concatemeric RNA, the next step in this temporal sequence, would then self-process to make viroids. And each viroid would then undergo independent replication the way viroids do it, by the production of rolling circle replication and then self-process. Go ahead.
Maybe methylation isn't perfect, because I think you mentioned like, or maybe for just a split moment it was not methylated, so if it can just produce even just one RNA transcript, then it will undergo its self-replication, and then it will amplify itself and
Yeah,
no longer.
exactly, that's exactly what's going on. All we needed to do was make one copy from that transgene. And after that, it was okay for that thing to be silenced by methylation. Because that one copy of the RNA that was transcribed processed itself into viroids, and each of those independent viroids then replicated themselves in the normal fashion that viroids do. So you have to sort of try to see this in this temporal sequence like this in order to truly understand what's going on. So before I go further, does anybody want to ask me any questions or ask me to clarify things?
Can you re-explain that quickly?
Okay, let's just go back to a picture. Try to picture in your mind the following events. And the question we're asking is, if the integrated copy is methylated, how is it possible that we still see viroids in these plants, in these transgenic plants? So that's our question. Now let's start at point one. The tDNA integrates in an unmethylated state because it's coming out of a bacterium after all. It gets transcribed, producing a long concatenameric RNA. In the third instant of time, that long concatenameric RNA self-processes. Each unit cuts itself out of the concatemer and circularizes. Perhaps as that process is occurring, as that self-process is occurring, DNA methylases are finding all of these CCG or GGCC motifs and beginning to methylate them. We'll talk more about how they found those sequences because that becomes the real issue here. But for the moment, just say, yeah, they must have found those sequences, otherwise we wouldn't have seen the integrated copy being methylated when we did the Southern analysis. But at the same time as those methylases are finding those GGCC motifs, those already processed viroids are undergoing rolling circle replication, making dozens of viroids from each circle, which in turn circularize, making dozens more, and so on and so on. And so, how is it possible that these plants that have a highly methylated copy of the tDNA, of the viroid DNA in their genome, are still producing a highly methylated copy of the tDNA in the genome? How is it possible they are still producing viroids? Because they no longer need that integrated copy. They are now independently replicating themselves. The viroids are now independently replicating themselves. Yeah.
So the viroids are never methylated.
Viroids are RNA. They will not get methylated.
Okay.
Anybody else want me to clarify something? All right, well, let's just look again at what are the conclusions that we can perhaps draw from this. It's not the one I wanted to do. Okay, well, let me just make some comments without having the drawings in front of me for the moment. Keep in mind, when we integrated a mutated copy of these viroids, the concatemer that had the little deletions in each unit, we still saw transcription, but we did not see methylation, and we did not see viroids. So it struck these people, it struck Bocanega's group, that it is the actual presence of the viroids that seems to be connected in some way to the methylation of the genomic copies. And in order to prove that, they took a plant that had the deleted version of the viroid genome so that it could only make long concatenameric RNA, and they superinfected that with viroids. And what do you think happened? The genomic copy got methylated by the introduction of those viroids. Now keep in mind, those viroids don't encode any proteins. So how is it possible that this RNA that they introduced is somehow able to direct methylation to this integrated version that could not get methylated on its own? Also keep in mind, we are talking about this single transgene. Other genes in the genome were not showing differences in methylation. So this is really different in many ways than the maintenance methylation that we've been talking about earlier. We are not simply getting global methylation. We are getting very specific methylation. And that specific methylation is totally under the control of some condition that, at least in our hands, doesn't seem to be dependent on the production of a unique protein. If you want, you could even, well, okay, no, let me not say that. Okay. I don't want to give out too much now. All right? So everybody kind of see what's going on with this experiment? And what people saw was that maybe this is a model for the general process of transgene silencing that we occasionally see when we make transgenic multicellular eukaryotes. The idea that evolved from this experiment was something like this. When that first transcript of the viroid was being expressed, it was just like 1 or 2 or 10, maybe even 100 copies of that messenger. But it didn't stay very long in the nucleus, and it got out into the cytoplasm and, you know, could have no effect on the genome that it was copied from. But in the case where the viroids are replicating, whether because we put a viroid into, you know, we co-infected with a viroid, or because the transcript itself self-processed into viroids, those viroids, as circular molecules, don't leave the nucleus. They just accumulate there. That may be part of the reason that plants infected with viroids show all sorts of growth inhibition. Their genome, or their nucleus, I mean, just gets clogged with all of these circular RNAs that do nothing purposeful. But one thing can happen. Those circles can go and base pair to the integrated copy that we inserted in the tDNA, because each of those viroid circles is homologous to the linear DNA molecules that we put into the genome. And the idea then is that perhaps we can think of it like this. It's really not the best picture. Did I have a better picture here? Okay. Well, you can picture it like this. The tDNA, the integrated copies of the viroid genome, started out unmethylated. As the circles accumulated, some of them would be able to base pair back to the templates that they were copied from. They would essentially form a triple helical structure in which the RNA intercalated between the two DNA copies. People picture that? Well, it turns out that triple helical structures like that, RNA DNA triple helices, are very attractive to another kind of DNA methylase that's called an RNA-dependent DNA methyltransferase. This is a unique enzyme that is not involved in maintenance methylation, but rather in gene-specific methylation. when it sees these triple helices, it's a warning, you might say, that things are not progressing in the normal fashion where RNA is made and exported, but rather the RNA is accumulating to sufficient amounts that no matter how weakly it binds back to its template, it is binding there and intercalating itself into the double helix, creating this triple helical structure. And then the RNA-dependent DNA methylase, methyltransferase, binds to that triple helix and methylates the cytidines in the vicinity of the triple helix that overlap the triple helix. Okay? Well, initially, this is only going to be the transcribed region of the gene because that's the only region of the gene that can base pair to an RNA that is present also in the RNA. But as the methyl groups are added to the transcribed DNA, sometimes that DNA methylase doesn't release immediately, but instead drifts upstream or it drifts downstream and continues to methylate sequences outside of the triple helix. And then over time, some of these DNA methylases, these RNA-dependent DNA methylases, drift all the way up into the promoter region and methylate cytidines up there. And when they do, what happens? They shut off transcription of the integrated gene. So it won't be instantaneous. It will take time for this process to occur. And as we can talk about later, we're talking about a lot of time. But once that DNA has been methylated as a triple helix, it will be passed on in a methylated state from generation to generation with the assistance of the maintenance methylases that just see a hemimethylated sequence and then, so like here, for example, we have the fully methylated state after this has happened, and then DNA replication occurs, and we now have hemimethylated sequences there, and then the maintenance methylase comes in and methylates the other strands. And so the methylation state of the DNA persists without any continued expression or absence of expression of the transgene. Yeah.
When you say inherited, do you mean meiotically or via reproductive offspring?
For the moment, it's all mitotic.
Yeah.
Things get complicated when we start talking about meiotic inheritance. So for the moment, like I kind of showed you in that mammalian diagram a week or two ago, there's a demethylation process during embryogenesis, and then it gets reimposed de novo. But as I hinted at also last time, there are certain genes that we don't fully understand the details, but you'll see some explanations later, which seem to remain methylated even after all the rest of the gene has been erased. And so that allows us to have an inherited epigenetic trait in certain cases. But let's not focus on those exceptional cases for the moment. Let's just focus on in general. During meiosis, DNA is demethylated. All the epigenetic traces, including ones like this, are lost. But what you'll see later, is they can get reimposed if the conditions that led to the accumulation of RNA persist. Of that gene-specific RNA persist. Any questions so far? Why do plants have these kinds of RNA-dependent DNA methylases? Well, the explanation seems to be that this is an anti-retrovirus defense. Now, remember that we have these retroviruses. These are viruses that get converted into DNA, integrate in the genome, and then continue to get expressed. We also have transposons that seem to have evolved from these retroviruses. they have lost the ability to excise and make a new virus, but they can jump from one location to another. And every time they jump, they're increasing their copy number and they're increasing the probability that they're going to inactivate an essential gene leading to the death of that organism. What can the plant do, especially with these retroviruses and these retrotransposons? They don't have any characteristic protein that the plant can make a defense against. And plants are not really good at making protein-specific defenses like mammals can, for example, or even many invertebrates can. So what can they do? They have to have a mechanism that detects the transcription of the gene in some way or another. And the most general way that we think this occurs is that when there is a burst of retrovirus activity, we're getting an accumulation of a lot of RNA, and then, as in the case of these viroids, some of that RNA can base pair back to the DNA template it came from, forming a triple helix, which would then attract the RNA-dependent DNA methylases, which would lead to the methylation of that sequence and eventually the shutting off of the gene that was producing that RNA. And in that way, the eukaryote can defend itself against excessive replication of these kinds of transposable elements. If we knock out the RNA-dependent DNA methylase, we see a burst of mutagenesis occurring, because now the plant or the animal, the Drosophila, for example, cannot shut down the transcription of those genes in any other way. And so they just keep copying themselves and hopping around from gene to gene until they eventually hit something critical, leading to a mutation of some kind. Any questions on that? I do want to emphasize a couple of points again. Viroids normally don't go through a DNA phase. This is totally an artifact of the experiment that was conducted. Therefore, this defense that I've just described does not work against viroids. But it does and can work against transgenes. If we happen to build a transgene, maybe with a good promoter, and by chance it integrates downstream from a strong enhancer, we might get far more gene expression than we expected. So much so that some of that transcript binds back to the template it came from, forming a triple helix, attracting the RNA-dependent DNA methylase, leading to the methylation of that gene and eventually to its silencing. Does every transgene get silenced? No. The estimates are around 1 in 8, maybe a little bit more in some organisms, and maybe 1 in 7 in other organisms. That means that, that in 1 of 8 or 1 in 7 cases, something happens to the transgene, possibly that it integrates like this, leading to its methylation and its shutoff. But we do see that in these cases, we frequently do see the fact that there is an increase in the methylated sequence of the promoter regions in transgenes that have been silenced, and not in transgenes of the exact same structure that were not silenced. So we do think there's a strong correlation there between the methylation state and the silencing of some, not all, but of some transgenes. All right. Well, I hope this sort of is, or at least it'll resolve itself into something that seems to make sense to everybody. But I do have to go on and just briefly introduce you to a different kind of silencing, which is post-transcriptional silencing. Yeah.
Before we move on, I have a question about this. So all the examples that you gave were about like genes that we introduced or that were introduced through like a viroid or a retrovirus or something. Does this type of silencing happen at all in like a natural or like wild type organisms?
there are cases where it seems to be happening. In those cases, what seems to happen is, and it may not be that common, is there's been a translocation event in which a gene has moved from its original location to a new location. And perhaps that has changed the level of expression of that gene and led to the methylation of that sequence. But I gotta say, I'm just trying to think back for a moment on some other experiments. I don't recall a clear example of this kind of silencing in any natural gene. The genes we call natural are always in their right place. They are in the regions where they will be expressed in a highly predictable fashion. They may be near methylated regions or not near methylated regions. They may have silencers and enhancers, or maybe they just have a few enhancers. But they're expressed at levels that don't lead to the excessive accumulation of RNA and the silencing of the genes. We had no clue that this kind of a process, or the next one that I'm going to talk about, ever existed until we started putting genes where they shouldn't be. And that was through non-homologous recombination events. Any other questions? I think I'm just going to stop here. I'm not going to introduce. What we'll do when you get back, so next week is break, right? You guys are lucky. So what we'll do when we get back is I'm going to talk to you about post-transcriptional gene silencing. And if you thought transcriptional gene silencing was a little kooky and weird and all that kind of stuff, wait till you hear about post-transcriptional silencing, because it is loony. But it is very, very, very much true and affects a lot of our transgenes that we put into the eukaryotes. So we'll talk about that stuff later. Okay. Hope you have a good break. I hope it's peaceful, and that you return restored, as all of us deserve to be. Okay. That's all for now. Oh, except that I do have to give you your tests. Shoot. You were just going to walk right out of here?
Maybe we don't want to